Hello, everyone. I think attendees are still trickling in, but I'll start. Welcome to another Connect Science seminar on this Thursday. And it's a very great pleasure to welcome Michael Ruppy to this seminar today, who joins us from the University of Berkeley. Before I introduce him, let me just touch on a few housekeeping items. For upcoming seminars and also past recordings, we'd like to remind people that you can go to the website of the seminar series, and there's a long list of outstanding seminars by many speakers and all the upcoming seminars. Also, for those who have not attended these seminars before, I'll kindly ask you to ask questions using the Q&A feature and not the chat window, which allows us to come back to most of the questions at the end, but if we're running out of time, we can also circle back questions to the speaker. So please use the Q&A feature. I also see quite a number of people joining from Berkeley, so you seem to be pretty popular out there. Let me get back and let me introduce Michael Ruppy, who, as I said, joins us from the University of Berkeley, where he is the Peter Hirt Chair of Cancer Biology and a professor in Cell and Developmental Biology. He also is an investigator of the Howard Hughes Medical Institute. And I'm just going to spend a few minutes to say a few words about his background, which otherwise would take way too much time of a seminar. He grew up in Germany and did his PhD at the Max Planck Institute with Stefan Jentsch, where I think his interest in ubiquitin and ubiquitin-related molecules started. Then did a postdoc with Mark Kirschner here at Harvard Medical School before moving out west to Berkeley in 2006. And he's since then really had seminal contributions to many areas, such as cell cycle control, many aspects of cell fate decisions. And one unifying theme around this is, I think, has always been understanding it from the view of ubiquitin, which, as some people may know, is very close to my heart. I don't want to speak much about his science because I think he's going to tell us more about it, and I'm sure he has some new, interesting, novel data that I'm very keen to see. He's received numerous prizes for his work, and I'm not going to name them. Most recently has been a National Blavatnik Laureate. He's received a Wilczek Prize and many others. And I think what's sometimes less well known to the field, he's also been a pioneer in targeting the ubiquitin system for drug discovery. He's a co-founder of Nurix, and it really goes back to pioneering work done in his lab many years back that I've followed closely, and I think it's great to see how this field is really contributing now to the development of novel therapeutics. I'll stop here. Misha, it's great to have you, and please take it away.
All right, can you hear me?
Yes.
Yep. Oh, super. Thanks, Eric, for the very kind introduction. Thanks for inviting me. It's awesome to give a talk in Boston in August and not sweating to death. I'm really grateful that you have me here. So I think these ideas of running science in this time of pandemic across the continents is really inspiring, and seen this not only between institutions as today, but also in my lab where many students pivoted to projects targeting COVID and really showing that science matters, which I think is what we need in these days. What I want to do today, though, is talk about two quite exciting areas in my lab that are new to us, that have nothing to do with COVID. They're really only targeting the core interest of my lab in understanding molecular mechanisms of cell fate specification. And specifically, I want to introduce you to two new quality control systems that we discovered. One about complex quality control, and the other one in homage to Bill Kaling's lab at the Dana Farber, a new pathway for redox control in cells. So all of our work really starts with ubiquitination, with ubiquitin, a small molecule which is highly conserved, absolutely essential, and performs so many important functions in the cell, allowing the cell to really control its fate very precisely. This modification is set into place by specific enzymes called E3 ligases, which recruit a substrate as well as an activated form of ubiquitin and can transfer ubiquitin, shown here in the red ball, to a substrate. This modification is then detected by effectors which practically couple ubiquitination to a downstream biological event. and thereby determine the outcome of this modification. And as anything reasonable in biology, this has to be reversible, and there are the so-called deubiquitinases or DUBs that can cleave it off. And one reason why we're so excited about this pathway is that there are so many of the human enzymes that are dedicated to either adding or removing ubiquitin. About 1% of all of our genes are really linked to this pathway. And so you have many opportunities to study how cells control their fate or their homeostasis. Another reason why we like ubiquitin so much is that it's a modification that comes in different flavors, and these different flavors often have distinct functions. So ubiquitin was first discovered as a single modification, a single molecule transferred to a histone protein that is called monoubiquitination, function that often changes protein interactions. But then pioneering work from Alex Warshawski, Dan Finley, close to where you are, and many others now, has shown that you can form polymeric forms of these ubiquitin modifications where one ubiquitin molecule is attached to another. If you use the same attachment site, you form so-called homotypic chains. And as Hermann Meyer initially showed in my lab many years ago now, if you modify a ubiquitin on two sites at the same time, you can build these branched structures. And again, these modifications can really differ in their output and so allow ubiquitin to cover a whole range of reactions in the cell. The best known function of this pathway, and the function that you will listen to today, is protein degradation, mostly through the proteasome, through homotypic K48 or these branched K11, K48 chains, but it can also target proteins to autophagy. Now it can change protein interactions, means to specific and reversible complex formation, or change activity, localization, many other features in the system. As expected for a pathway that's so intriguing, it's very important in development. If you lose certain ubiquitination enzymes, such as the one that really got me hooked the most, the anaphase promoting complex co-discovered by Mark Kirschner around your corners, you do not divide, you do not differentiate. And there are many other E3 ligases that play central roles in differentiation, as the colon-3 enzymes you'll hear about today, or in cell survival, for example. Now, from a clinical point of view, one thing that has become really exciting is the recognition that misregulation of these pathways leads to many severe diseases. With respect to cancer specifically, we know now that I think out of the top 7 tumor suppressors mutated across all cancers, 3 actually are E3 ubiquitin ligases. And some of the nastiest oncogenes, such as MDM2, also fall into this class of enzymes. But what made this really exciting for us was the recognition that you can target the system now, not only to inhibit a badly behaving enzyme, but you can also redirect these signaling machineries to pathological proteins to change their behavior. This really is founded on groundbreaking work by Bill Kaelin, who taught us how thalidomide works, Jay Bradner at your institution when he was still there to show how you can build actually a PROTAC, and then lots of amazing work by Eric Fisher and Nathaniel Gray. We contributed to this field, as Eric mentioned in the introduction, by developing this idea that you can also make molecular glues. These are simpler molecules, only one entity, that can sandwich between an E3 ligase and bridge over to a pathological substrate, and in this case, induce degradation of a protein that would cause colon cancer. So understanding ubiquitin is really important by itself for knowing how the cell regulates important behaviors, also for knowing which enzymes to select for drug discovery, but we really use it mostly as an entry point into figuring out how these reactions then control cell fate specification, both in development, how you become a certain cell type, as well in disease, for example, how you maintain cell integrity over life. And so today I want to highlight 2 aspects of this work. The first is really our discovery of a new type of quality control, which we call quality control of complex composition. That is the ability of cells to detect complexes that simply have the wrong subunit. We all know from life you shouldn't partner up with bad people. Same is true in cells, and cells can detect those bad pairings even if there is no misfolding. And I will show you the mechanistic underpinnings of this quality control pathway. And then the second part will be a new stress response we very recently discovered called the reductive stress response. That is very close to the hypoxic and oxidative stress responses. As you'll see, it's a way to control the output of reactive oxygen species, and in the course of this, maintain stem cell integrity. So we'll start with the first project, dimerization quality control. Every project in my lab really starts with a curious mind, with a courageous student that is not afraid of venturing out into the unknown. And this is very much the case in this project, where I had the immense luck of recruiting Elijah Mina as a student to my lab. Elijah discovered what I'll talk about in a minute, the pathway, he reconstituted it, and we'll show you also the molecular understanding, atomic understanding of this pathway. Elijah is now a postdoc in Steve Elledge's lab across the road from you guys. And so if you have questions, you can go right to the source where it all began. So Elijah's graduate work in my lab rested on the work of other students, Colleen McGurty and Lin Yang Ying, as well as a postdoc, Achim Werner. And what these three wonderful people have managed to do is that they found a really important role for ubiquitin in craniofacial development, which is a very intricate developmental program. And we found that there is one of these E3 ligases, the specificity components of the system, called Cullen-3 KBDBD8. And I'll show you in a second what that letter jungle actually means. This enzyme is important so that stem cells can specify into a neural crest fade. These neural crest cells differentiate further, and then you need a different flavor of this ligase, Cullen-3, paired with a different adapter called Calc12 to make downstream cell types, so-called chondrocytes, being able to secrete collagen, which functions as a blueprint for bone formation in the cell. Now, about 30% of all birth defects in humans are actually caused by problems in craniofacial development. So this is an incredibly important pathway. And so we thought that it might be quite critical for an organism to not only make sure that these reactions happen, but also control the regulators themselves. And so Elijah's job was figuring out to learn how these BTB proteins or these Cullen-3 E3 ligases become active at the right time and the right place in a developing organism. Now, he focused initially on this E3 ligase called Cullen-3 KBDBD8, which controls the first step of neural crest specification. This is how this enzyme looks like. It has a scaffold, which gives it its name as a Cullen-3 ligase. It has an active site centered around a ring finger protein, RBX1, and then it uses so-called BTB proteins as substrate adapters. These were actually co-discovered by Wade Harper, who is very close to you guys. KBDBD8 is one of those BTB proteins. It uses its BTB domain to both bind to the Cullen-3 backbone as well as to homodimerize. And this homodimerization is absolutely essential for its function because it allows it to read out multiple degrons in a substrate. What it actually does is it doesn't degrade the substrate, it monoubiquitinates its substrates. This leads in a very intriguing way to biogenesis of novel ribosomes that have different specificities. They like to read out mRNAs that code for proteins for neural crest fade, while it doesn't like to translate proteins that will lead to a different telencephalon fate during development. Now, we were interested in this because not only is this really interesting biology, but it's also medically highly relevant. If you have mutations in the substrate that remove the E3 binding site, these mutations account for about 95% of cases of a disease called Treacher-Collins syndrome. This is a disease of aberrant craniofacial development due to a lack of neural crest specification. Conversely, if you have too much of KBDBD8, this came out of a sequencing study in Australia a few years ago. You actually get too many or the wrong type of neural crest cell, and that leads to a cancer called melanoma. And the beautiful work by Len Son around the corner where you are showed that melanoma is in fact a neural crest-derived cancer. So too little causes developmental effects of this enzyme, too much causes cancer. So we really wanted to know how this is controlled. And so Elijah used a proteomic, before going to this, I should mention again that this KBDBD8 is a member of the BTB family of proteins. We have about 220 of those in our genome. So this is a very abundant protein family. About 100 fall into classes of E3 ligase subunits. So they define the substrate specificity of this Cullen-3 enzyme. And for all of these, I think homodimerization is an essential aspect of their function. And the same holds true for about another 100 BTB proteins that are transcription factors, including some very important for tumorigenesis, such as BCL6, where again, the BTB domain allows some binding to an effector, but also performs an essential function in homodimerization. And so what I'll talk about is really a system that makes sure that you form these functional homodimers, but avoid pairing with the wrong BTB protein, which could be easily happening because there are so many of these variants that could pair in the wrong way. And so Elijah had used initially, and that is published now about a year ago, has used a proteomic approach to screen for regulators of BTB proteins and found a really interesting regulator, which was an E3 ligase by itself. It's a ligase called SCF-FBXL17, where you have a colon backbone. And I should say that these SCF ligases themselves were discovered by Steve Elledge, where Elijah is now working in the lab, as well as Wade Harper in Harvard. Now, they have this colon 1 backbone. They have the active site where ubiquitin transfer takes place. And in this case, they use a so-called F-box protein to recruit substrates. And what was amazing right from the get-go was our realization that this FBXL17 protein doesn't recognize a single BTB protein, but it can basically bind almost all of them. And it will polyubiquitinate them and trigger degradation of these proteins. So this is, from the get-go, a new class of regulator that controls a very widespread domain rather than a single substrate. Now, of course, we wanted to know what the specificity is. You can't just degrade all BTB proteins. No organism would survive this, so this had to be highly regulated. And through, sometimes very hard reconstitution, and I really pay tribute here to Elijah, who went for about a year through experiments where everything failed that he did. But it was important to lead him to the insight, what really is at the heart of this. So the persistence here and his ingenuity in building the system is really amazing. Now, I'll just show you the one figure that summarizes this, like 1.5 years of reconstitution work. As I mentioned, these BTB proteins act as homodimers, and Elijah found a way how he could connect to BTB domains so that he can build homodimers of defined composition. This system also allowed him to build heterodimers, which are not active in the cell, but which we detect quite abundantly when we do mass spec experiments. And what is also intriguing for us, or was intriguing, is that these dimers, homo versus heterodimers, do not differ in stability. Both are very, very stable complexes in the nano or even sometimes picomolar affinity range. So they don't just fall apart by themselves. When he then when he built now these complexes of defined composition and looked for binding to FBXL17, here's inputs. These will be the pull downs, where you have a homodimer on the left side, another homodimer on the right side, and then the pairing, the heterodimer resulting from combining them in the middle. You'll see that independently of what BTB domain we used, and Elijah made more than 75, I think, at the end of the day, it always bound the heterodimer, but not the homodimer. And so this led us to the realization that this is a quality control enzyme that can distinguish complexes based on composition. The active homodimeric complex would not be recognized. It would be kept intact and it could perform its function in signaling, while the inactive and potentially toxic and very stable heterodimeric complex would be recognized selectively by FBXL17, which would then degrade this complex to make sure that the cell only has functional BTB proteins. And we found through work in xenopus development that this was particularly important both in the peripheral nervous system, where the neural crest is, and this is really through controlling KBTBD8, but also in the central nervous system, particularly the development of Rowan beard neurons, which sense touch in the developing xenopus embryo. So this is a really intriguing activity in cells, and of course there are thousands of biological questions that come up right away, but one of the most striking questions, in fact, was a biochemical one. And this is, you have an enzyme, FBXL17, which has to specifically detect its substrates. And now it has, we have about 200 BTB domains, so you can form these functional homodimers. And if you combine them just randomly, stochastically, you could, in theory, form about 20,000 inactive heterodimers. And so this enzyme must be able, first of all, to recognize 20,000 substrates, which is amazing. How can you have specificity when you recognize so many targets? And then compounding this problem is that in some cases, the subunit that's present in a heterodimer is just the same that would be present in the homodimer, yet this enzyme still has to be able to distinguish those complexes. And we realized that in order to address these questions, how is dimerization quality control really working? We needed to finally succumb to the fact and follow Eric's footsteps and learn structural biology. And this is really what Elijah did in an amazing way. Initially, we decided to go by cryo-EM, and I have to say that Elijah got fantastic help by Basil Gerber in Eva Nogales' lab at UC Berkeley in this work. He purified a almost full-length colon 1, skip1, FBXL17, E3 ubiquitin ligase, and then he produced a full-length substrate. In this case, we used KEAP1. This is one of these very frequently mutated tumor suppressors, and you'll hear more of it in the second part of my talk. The little trick that we could use here is that we found a point mutation in KEAP1 that behaved just like the heterodimeric complex. So we could make a dimeric complex, was very stable, nanomolar, single-digit nanomolar affinity, didn't fall apart by itself, was a dimer, no monomers ever detected, but behaved as a substrate of this dimerization quality control. We could mix them, we could purify the complex, cross-link it to stabilize interactions, and then we got an about 8 angstrom cryo-EM structure of this full-length substrate bound to an almost full-length E3 ligase enzyme. And this actually had a few really intriguing and exciting findings. The first is that FBXL17, shown here in orange, the substrate specificity subunit, found the PTB domain of KEAP1, which is expected for an enzyme that monitors dimerization through this PTB domain. That was cool. That confirmed a lot of the biochemistry we had just published. The second cool finding I want to mention, because work by Eric and Nathaniel in your institution really is in developing degrader molecules. And often when we build small molecule degraders for therapeutic benefit, we worry about lysine residues being close to where the pathological protein would be bound by the E3 ligase. In this case, substrate specificity is determined in one substrate domain, the BTB domain, but catalysis will occur in another domain in the Kelch repeats of KEAP1, suggesting that binding and ubiquitination could actually occur in quite different domains that you don't have to worry too much about lysines. But of course, out of all of the findings, we had the most striking one was we put in a dimeric, very stable dimeric complex into the reaction, but when we solved the structure, there was only one molecule of KEAP1 left. And this suggested that maybe one aspect of specificity here is that this machinery can go after complexes that either fall apart more often or can be split apart more easily than the homodimeric counterparts. Now the resolution was not high enough to really do mechanistic work, so Elijah bit the bullet and learned X-ray crystallography. He brought this to the lab, and I'm eternally grateful for Elijah to doing this. He could not have done this without John Kurian's lab, in particular Christine Guy in John Kurian's lab, who really was inspirational in how they opened the doors to allow us to play with crystallography. Elijah purified just the specificity component, which is this F-box protein, as well as the BTB domain. Again, he went for this mutant BTB domain of KEAP1, which we could easily study. This is, again, a dimer. Nanomolar stability doesn't really fall about all that much when you incubate them, even in a 24-hour chase, actually. He mixed these two, purified the complex by gel filtration, and then got about a 3 angstrom X-ray structure, again, of an E3 ligase bound to a folded substrate, not just a peptide. And this is how the complex looks from the side, and this is how it looks from the top. And there are a few, again, highlights of the structure. These FBXL17, these FBXL proteins are leucine-rich repeat proteins. You have here the leucine-rich repeats. It's actually longer in FBXL17 than in other FBXL kind of proteins. And then it's further extended at the C-terminus through this helix, which we call the C-terminal helix, or CTH. And through this like solenoid structure and this helix, what this ligase does, it really just circumvents, it hugs the BTB protein. And in fact, what happens is that it can hug proteins that have the right shape, whereas if you have little bumps there that would expand the structure, you would not be able to bind. And so this suggested that the first kind of specificity in the system is actually not a sequence. It's not a dagron, a simple amino acid sequence, but that this machinery recognizes the shape of its substrates, which then allows it to recognize hundreds of proteins which just look alike. The second insight that we got out of the structure was that it explained immediately why it binds monomeric BTB proteins. And blue here is a BTB one subunit. Normally it forms the dimer by binding to a second subunit, and you can see this C-terminal helix actually occupies exactly that binding site. So you can either form a dimer or you form the complex with the E3 ligase. This helix is connected to the rest of the ligase through a very flexible and long linker, and we believe that initially this is actually looped out so that you can recognize the dimer, and that this then probes the stability of this interface, occupies it, and then locks the system in place for ubiquitination. In fact, we think that this is more active in disrupting complexes than just capturing monomers. And that comes from a beautiful biochemical assay that Elijah built based on a lot of work that people like Eric has done, or Nico Tomei in Basel. Elijah purified substrate BTB domains where one subunit is labeled with one fluorophore, the other subunit is labeled with the other fluorophore. We placed them so that they're close enough to engage in fluorescence energy transfer. And so when you form a dimer, this donor fluorescence will actually be quenched. Now, if FBXL17 disrupts this and a subunit is kicked out, this quenching effect will be lost and you see an increase in fluorescence at the donor site. And so we could ask, is this just happening by chance, or is this happening in a more active process? And it's actually a really beautiful reaction. If we add FBXL17, we see this increase in donor fluorescence, showing you that we lose dimers. But if we use an FBXL17 variant that lacks the C-terminal helix, which occupies the site which we think is the pincher, we don't see this happening all that much. And what was equally important, if we use a sink, a very large excess of an unlabeled BTB protein that could capture any monomer that would spontaneously dissociate and take it out of the equation, that doesn't have an effect. And this shows that there's at least an active contribution in disrupting dimers, while there might be a minor contribution in soaking up spontaneously dissociating complex subunits as well. So what this suggests is that this machinery, this E3 ligase, has two functions, not only ubiquitinations, but it also probes the stability of the complex. And this then leaves us with the last question I briefly want to address in this part of the talk. And this is how can you now decide homodimers from heterodimers? Do the structures tell us something about this? And really the key to this, again, came out of a set of structures Elijah solved. He got these mutant substrate BTB domains solved. And just like the wild type, it looks like the wild type BTB domains and has one really intriguing feature. And this is that the N-terminus of the BTB domain reaches out to the other subunit. And it's hard to see here, but it forms a beta sheet between beta strand of one subunit and a beta strand of the other subunit, an intermolecular beta sheet. When you're bound by the E3 ligase, though, this has to be broken and it folds back onto its intramolecular beta sheet structure. So clearly, this is sort of a gatekeeper for recognition by FBXL17. You have to release it in order to be able to bind to the substrate. And what was exciting is when we generated models of heterodimeric structures of two BTBs, we saw that clashes really occur only here at this beta strand structure, as well as residues next to this beta strand, which we call the hinge, suggesting that heterodimers might not be able to place this beta strand in this intermolecular position and might have it more in an open position as shown here. And this raised the possibility that the position of this beta strand actually gates access to FBXL17. We could test this quite easily. We force one into place through a simple fusion, but then we looked at the other one, whether it's open or closed. And what we did is we basically replaced the clashing residues of one subunit with the residues that would be present in the other subunit. And so with this little swap, what we accomplish is we make a heterodimer that looks like a heterodimer only in this intermolecular beta sheet. And what is striking is that the normal heterodimer is recognized by the ligase. The homodimer is not recognized, as it should. And the swap, where we make the heterodimer look like a homodimer just there at this one very short 5-amino acid beta sheet, that escapes recognition by the machinery. What makes this even more exciting is when you look into evolution, this beta strand is the most rapidly evolving sequence in BTB domains, together with some residues that actually touch the beta strand, as you would expect. In fact, out of the 200 BTB proteins in our genome, there is not a single one that contains the same 5-amino acid beta strand. So we think this is actually nothing else than a barcode for dimerization. Homodimers will have a perfectly matched barcode. They can form this closed conformation, whereas heterodimers have clashes. They will release this at least more frequently, and that makes them accessible to FBXL17. So the third part of specificity here comes from complementarity that is defined by a rapidly evolving barcode for dimerization. So to summarize this first part, homodimers can occupy most of the time this closed conformation. In this conformation, FBXL17 does not recognize them, and so they are stable. Heterodimers have clashes here. They release it into the open conformation. That allows FBXL17 to recognize. We know initially it's the dimer. It then probably through some allosteric means further destabilizes this interface, leading to dissociation of one subunit, stable binding, and ubiquitination of the other. And of course, the monomeric subunit that occurs here will rapidly be picked up by another FBXL17 molecule to degrade. This is an active mechanism of complex disruption. We don't by no means exclude the possibility that you can form unstable heterodimers that fall apart by chance and can be picked up by FBXL17 as well. So there are a few general lessons we can learn from this work by Elijah. One is that, again, I just want to repeat for PROTAC and small molecule degrader development, we don't have to worry all that much whether there are lysine residues close to the binding site. What we have to worry about is whether there are lysine residues that are accessible to the active site of the E3, and the structure, I think, where detection as well as ubiquitination occur in different domains of a substrate suggests that this is not the same thing. We also learned that specificity doesn't necessarily, is not necessarily encoded by short sequence motifs or degrons, which is often the case, but not always. In this case, it's a compilation of events such as detecting the shape of BDB domains, complementarity through this intermolecular beta sheet, and then of course also stability of their interactions. And for us, this is really cool. We're really excited about this. This opened a new area of biology in my lab. If you're interested in this, reach out to us. Many opportunities here. There are many of these recurrent interaction motifs that have to form either very specific complexes, and we think that this mechanism we discovered here is very prevalent in biology. We have found examples of this, and we're now studying them to see whether mechanistic details look similar as well. All right, this was one example of how this machinery can control in a very unique and cool way very important regulators of development. Probably at least equally exciting is the second part, which I chose to talk about today because Dana Farber is really the birthplace of the hypoxic stress response due to the beautiful work of Bill Kaelin. And this is something that comes very close, I believe, the reductive stress response that we very recently discovered that is also essential for maintaining the levels, physiological levels of reactive oxygen species in cells. And this is work by, again, wonderful people, a postdoc in the lab, Andrew Manford, who came with the idea of looking into a particular biology that we had not worked before, brought it to the lab, established it, and then luckily it paid off by giving him a gold mine of findings. And he was helped in fantastic ways by a graduate student, Fernando Rodriguez Perez. Now, the question really at the heart of this is, again, a developmental question. We know that during development, an organism, an embryo, very often experiences stress that can be nutrient limitations, problems in chemical exposures, temperatures, and so forth. And what the cells of these embryos then accomplish is that they detect the stress through sensors of so-called stress responses. And then they set in motion reactions that basically alleviate the stress, counteract it, so that organismal homeostasis can be maintained. This is best investigated in protein quality control and preventing misfolding, where we know that if we lose the stress response pathways, we would accumulate aggregates, and that is very often linked to neurodegenerative disease. But as these pathways became better understood, we also learned that it's not only important to maintain tissue integrity. Very often deletions in mouse models, for example, led to early developmental phenotypes, suggesting them that really stress signaling is critical for early development and tissue formation, and we wanted to understand how that works. A pathway which really illustrates all of these, and which is very important for this talk, is the very heavily studied oxidative stress response. It's dangerous for cells to have too many reactive oxygen species because they could oxidize proteins, nucleotides, lipids, and so forth, and that causes many, many problems. So if that happens, the cell actually inhibits an E3 ligase, again, of this colon-3 family, and in fact, it's the same substrate adapter that I showed you the structures of KEAP1. If this is inhibited, that allows the accumulation of a transcription factor called NRF2. So this is the sensing event, this is the effector event of the stress response, and this induces expression of antioxidant proteins so that you can bring reactive oxygen species back down to a normal level. We know that if you have problems in this pathway, there are many consequences. The most dramatic to us these days is that mutations in KEAP1 are the most frequent event in lung adenocarcinoma, and it's very often seen in mutant RAS metastatic lung cancer as well. The renal cancer is affected by this pathway as well as the survival of certain neurons. But what has been clear when KEAP1 had been deleted in mice is that lack of KEAP1 also leads to a perinatal death phenotype due to problems in esophagus formation. So there is a developmental effect as well. We didn't necessarily want to study a pathway like this with our questions because there are so many people doing fantastic work there already. So Andrew took the slightly harder route to basically first discovering his own stress response that he could then study in this context. And the pathway in which he studied this, the developmental context, is myogenesis or muscle formation. There are many advantages to this. It's a pathway that's actually in its basics fairly well understood. And we can reconstitute this in vitro very easily using muscle stem cells or myoblast precursors. But most importantly for his work, it's a pathway that's very sensitive to stress. You all know, if in earlier lives, we went to the gym, now we go running outside. If you have exercise, that increases myogenesis, hopefully, if you do it right. If you eat very badly, if you eat wrong, if you fast, that has the opposite effect. Nutritional stress can prevent this from happening. Actually, a problem also in late stage cancer as well. And so basically, Andrew's idea was, can we find regulators of myogenesis that might have an interesting stress function? And so he set up a RNAi screen focused on the ubiquitin system. And it was very exciting to see for myogenesis. This is just the efficiency of myotube formation in vitro. And it was really exciting for us to see that depletion of KEAP1, this oxidative stress sensor, completely eradicated formation of myotubes. So the green here are the myotubes, and you do not see them at all if KEAP1 was missing. So if you deplete KEAP1, so if you have the oxidative stress, you transiently inhibit it, but if you deplete it, you do something quite different. You persistently stabilize this transcription factor NRF2 that leads to persistent production, overproduction of antioxidant enzymes that, for example, make glutathione. This then not only binds the reactive oxygen species that are abundant, but it gets all of them, including the physiologically important ones. And actually in 1987, so that's more than 30 years ago, it had been recognized that the lack of reactive oxygen species is toxic. And this imposes a stress in itself, which is called reductive stress. So in a weird way, if you consistently misregulate the oxidative stress response, you impose another stress called reductive stress. And this reductive stress really is key to this mechanism we have here. We can show this genetically quite easily. If you don't have KEAP1, you don't get myotubes. If this is due to stabilization of NRF2 or producing too many enzymes of glutathione production, for example, co-depletion of these enzymes together with KEAP1 should rescue this phenotype. And that's, in fact, what we see for NRF2, as well as for 2 enzymes of glutathione production, showing you that persistent reductive stress actually inhibits myogenesis. We know this now also because we can take chemicals and we can scavenge reactive oxygen species directly, and that also prevents myogenesis very efficiently. So just a little bit of housekeeping definitions. Oxidative stress has been studied for many, many years. This is too many reactive oxygen species, clearly dangerous for the cells. Reductive stress has been around for a long time. We now know that this depletes reactive oxygen species that are important for signaling. We don't fully understand what they do in signaling. Something it's involved in growth factor signaling. We believe that the work coming that you're seeing in a minute also suggests that it's a sort of quality control signal for mitochondria. You can exert reductive stress by many, many means, mutations in KEAP1, for example, or other proteins, aberrant glycolysis, inhibition of KEAP1 through a lot of electrophiles. So there are many ways to get this reductive stress to experience it. And if you are not able to manage it, if there is no reductive stress response, you end up in cardiomyopathy, diabetes, and through KEAP1 mutations, clearly also in lung cancer. And so we asked whether this is a central stress. This is really difficult for cells to deal with. So if this is something that is bad, typically the cell has evolved a response. And so we wanted to know whether there is a reductive stress response that can detect there are not enough reactive oxygen species around and alleviate that condition. And we were lucky because our myogenesis results gave us with a genetic screening system to really identify those components. So just very simply, if we don't have KEAP1 in a myoblast, we don't form myotubes because we have reductive stress. If this is due to signaling through the reductive stress response, if we then would co-deplete a component of this response so that we shut this down, we should again rescue myotube formation. So we did this in a larger screen, a genetic modifier screen, and we got a really nice hit out of that, E3 ligase called FEM1B. This is just from the screening plate. You see suddenly here, we get myotubes again in a very efficient way. And we were excited about this because this FEM1B had popped up in our initial screen with completely the opposite phenotype as loss of KEAP1. Rather than losing myotubes, we got many more myotubes if this protein was depleted. So oxidative stress response and a candidate reductive stress response regulator were at opposite ends of this screen, which made a lot of sense. And just to show you the data, it's really dramatic. If we lose FEM1B, we get a huge stimulation in myotube formation. If we lose KEAP1, this regulator of oxidative stress, we don't get any myotubes. If we co-deplete them, we rescue both phenotypes to exactly more or less the efficiency of control cells. And so it really suggests that oxidative and reductive stress signaling balance each other and are very intricately connected with each other. And so together with many other experiments we did, it suggested that there's really a tight crosstalk between these stress pathways. And we now had the first candidate component of this. And again, it turned out to be an E3 ligase. In fact, colon-2 femon B is a so-called colon-2 ligase. So it has a colon-2 backbone. We had colon-3 and colon-1 before. This is just another one of those. Again, it's the scaffold. This is the catalytic center of this enzyme where ubiquitin transfer would take place. And femon B, again, is the substrate adapter that recruits a specific substrate. And we know that these enzymes, in large part actually through work at Dana Farber and Bill Kalin's lab, drive proteasomal degradation of their substrates. And so key to understanding whether this is a component of reductive stress signaling and how this would be occurring, we of course needed to find the substrate. And so we do this by mass spec, following compass mass spec approaches that have been spearheaded by Steve Giggy and Wade Harper across the street. In this case, we always have to make a substrate trap. If you just express the subunit itself, it would degrade the substrate. We set a point mutation so that it cannot bind to the rest of the ligase. It still can bind to the substrate, and so it's sort of trapped in this complex. We do label-free quantitative mass spectrometry, and we get the key substrate or the candidate substrates here in the blood. And the one that really brought our attention was this protein FNP1. You'll see that in the input side here, if we induce the ligase, FNP1 goes away, and we know that this is due to proteasomal degradation. Through a years-long process, we were able to get one point mutant in the ligase, which didn't bind the substrate anymore. It turns out to be a cysteine, and you might want to remember this. We're dealing with redox stress here. If you express this, actually the substrate is not degraded, and you can see it's also not bound, while it's nicely bound by the wild type. And this was the substrate trap that we used. It actually stabilizes the substrate and it's bound more efficiently than the wild type protein does. And actually, we usually get a list of candidate substrates, and we then have to perform a genetic screen to figure out which one is the important one. And it turned out to be FNP1. Again, we can show this genetically. If you lose this ligase, you get more myotubes. If this is because you accumulate a particular substrate, if you co-deplete the substrate in this background, you should bring it back to normal levels. And that's happening for FNP1, but not for the other candidate substrates that we found. So accumulation of FNP1 really accounted for this phenotype, and so identifies this protein as the key substrate. FNP1 is not a transcription factor as in the oxidative stress response. It's a metabolic regulator. Beautiful work by David Sabatini's lab at MIT showed that it can regulate mTORC signaling. There's also some indication that it binds the AMP kinase. Neither one of these two functions is actually important for what we are talking about today, but it is, from the outset, it was shown to be a metabolic regulator. I should say that it is recruited to mitochondria when they are undergoing mitophagy, as Wade Harper had shown, and this is much closer to what we're going to talk about in a minute. So the next step, though, is how is this regulated? In the case of the oxidative stress response, you have too many reactive oxygen species. That leads to oxidation of cysteine residues in KEAP1 that, by some still very poorly understood mechanism, lead to stabilization of NRF2 and antioxidant signaling. And so something must have happened here to turn on the ligase or to make it recognize the substrate when you have reductive stress. And so in order to do this, we have to find the sequence in the substrates that recognized by the ligase. We do this by very, boring kind of deletion analyses, not boring, but tedious, done by Joe Shi and Karen Shi in my lab, two undergrads that we had for a long time. They were able to identify an essential sequence here that was required for binding of the substrate to the E3 ligase as well as for its degradation. And then to see whether that's really a decron, a transferable recognition motif for ligase, we stole a page out of Steve Ellich's book and built one of those GPS sensors where we fuse the decron to GFP, and then we co-express it with mCherry, and then you can use FACS to determine the GFP to mCherry ratio. If this is mediating degradation, it will destabilize GFP and you get less green over red. And so very nicely for us in green here, the green curve is the green to red ratio in wild type cells. Normal cells, if we induce the ligase, as you can see, we move to the left because now we degrade this fusion protein. If we have the substrate binding mutant or the substrate trap, we actually don't see this degradation, showing that this is a specific decron. Even nicer is that if we delete femon B using CRISPR-Cas9, we can see that the curve shifts to the right because now you have more GFP. And that tells us that about 80% of NIP1 actually is stable in cells, and about 20% can be degraded under normal conditions as we mimic them, at least in the culture dish. Now we have the degron. We looked at it, and then it was very clear that we had fallen into something really cool, because there are just 3 residues in this degron which are absolutely conserved and invariant, and all of these residues are cysteines. In fact, Elijah, on his way out together with Andrew, was able to solve the crystal structure. I should probably show you first. These cysteines are absolutely essential. If we have the normal degron, this is the green to red ratio. If we induce the ligase, we shift it to the left, it's degraded. If we use it by mutation of these cysteine residues, first of all, it's shifted to the right to begin with because it's stabilized, but now expression of the ligase doesn't have any effect at all. And as I mentioned, Elijah was able to solve the crystal structure of this complex as well, and it showed a very novel way of interaction between a substrate and an E3 ligase. There is very little contact between amino acids, as he normally does, but the 3 cysteine residues of the substrate pair up with this one cysteine that we had found before in the ligase to chelate 2 zinc ions. And the zinc really is the molecular glue that holds this complex together and provides most of the affinity of this complex. What was also nice to see is these residues here, these cysteines, are actually pretty close to each other, and we know that in normal cells they are oxidized under normal conditions, and they form intramolecular disulfide bonds. And of course, if you do this, if you form an intramolecular disulfide bond, you cannot chelate the zinc, and so you cannot bind, the substrate cannot bind the E3 ligase. That immediately suggested that oxidation, which we see under normal growth conditions, should prevent binding of the substrate to the ligase, whereas reduction during reductive stress will allow you to form this interface and form the complex, as highlighted here. So of course, you want to see this. If this is correct, you would predict that reversible oxidation should gate substrate recognition in this case. We can do this by various means. The nicest one for me is a simple fluorescence polarization assay, where we take the degron, just the triangle here. We are first good biochemists. We take it up in a buffer with reducing agent, and we see that it binds the ligase with an affinity of around 80 to 100 nanomolars. Then for a minute, we forget our training and we take up the peptide in a buffer without reducing agent. If you leave it without reducing agent just for about 30 seconds or so, you do not see any binding anymore. You can then take these samples, you add reducing agent back, and you can completely rescue the interaction. So reversible oxidation in this case gates access of the substrate to the E3 ligase. We see the same in cells. I told you that about 20% of NIP1 is typically degraded if we impose oxidative stress, which we actually do by just ramping up mitochondrial function rather than by adding any bad toxin. We see that we get some stabilization, yet if we impose reductive stress, either through KEAP1 inhibition, which led us to the discovery of this pathway, or by just ramping down mitochondrial function, we now see induced degradation of this system. And we see the same for endogenous fooling of NIP1 as well. So we now have this sensing event. If you have reductive stress, cysteine residues in the degron of NIP1 become reduced. That allows it to form a very unique interface with an E3 ligase mediated by 2 molecular glue zinc ions, and that leads to degradation of NIP1. And so the last thing for this stress response then was to figure out what is the effect here, how do you translate degradation of NIP1 into production of reactive oxygen species to counteract the stress. And to cut a long story very short, this all boils down to the one organelle in cells that produce most of reactive oxygen species. These are mitochondria. And you really see it beautifully just by looking at them in electron microscopy. Normally, this is the textbook image of mitochondria, the bean. If we deplete the substrate, which would mimic degradation of NIP1, we actually get bigger mitochondria that seem to be more active in some ways, but this is not in any way exciting. What is exciting is what happens if we stabilize the substrate, because then all mitochondria suddenly turn pitch black. And this is a phenotype that was first discovered in the 60s, and it's called matrix condensation. And it suggests that these mitochondria have run out of substrate for oxidative phosphorylation. Supporting this is that many mitochondria also obtain this onion-like structure where they overproduce cristae. This is an attempt of these mitochondria to compensate for the reduction in oxidative phosphorylation, but just making more complexes of the electron transport chain. And then following up on the beautiful work of Wade Harper in mitophagy, if you have problems in the membrane potential or in oxidative phosphorylation, you induce degradation through autophagy, and we can see this in some cases starting when we stabilize NIP1. We know that all of this is just because you accumulate this one substrate NIP1, because you can all quantitatively rescue it by co-depletion of NIP1. You can see the same when you look at the output in reactive oxygen species. This is really what we care about in this pathway. If we stabilize this NIP1 protein, we shut down mitochondria. We don't see reactive oxygen species. If we get rid of this one substrate, we completely rescue them. And we have indications that degradation enough here of NIP1 enough is actually essential, sufficient to increase output through the oxidative phosphorylation cascade. And we don't know really a lot about how this works, but I think metabolomic studies give us some insight, which we have done together with Dennis Tito and Dan Nomura at Berkeley. We found that if you deplete NIP1, so you basically degrade this protein during reductive stress, you accumulate, shown in yellow here, a lot of metabolites that are used by cells to shuttle either redox equivalents or metabolites themselves into mitochondria, so that this TCA cycle can produce substrate for oxidative phosphorylation. When you deplete FEM1B, so you increase the levels of NIP1, you actually turn those down. So we think that NIP1 stability is a gatekeeper that allows the cytoplasm to produce or import metabolics, intermediates into mitochondria, so that you now can run the TCA cycle, produce substrate for oxidative phosphorylation, and in the course of that, produce ROS. So that's how the reductive stress works, the reductive stress response. We have a sensor which has these highly, highly reactive cysteine residues in its degron. They typically form intramolecular disulfide bonds. If you have reductive stress, these are reduced. This allows the substrate to bind the ligase, leading to degradation. This protein is a gatekeeper for metabolite import into mitochondria. So if you would stabilize it, which would happen under oxidative stress, you shut down mitochondria, so you produce less reactive oxygen species. If you degrade it, you turn on mitochondria because you give them substrate or fuel to run on. And that activation leads to production of reactive oxygen species that counteract the stress. So we think this is actually a rheostat in reductive stress. It acts by degrading FNIP1 and activation of mitochondria, which then produce the ROS to counteract reductive stress. But if you have oxidative stress, at least due to a higher load of metabolism, not due to damage in the mitochondria, that could lead to FNIP1 stabilization and inhibition of mitochondrial metabolite import, which would shut down those mitochondria so that, again, you can come down to normal mitochondrial levels. So we discovered what we think is a system to tune mitochondrial output. The most striking thing, and this is what I want to leave you with, is that this is now a third pillar of sort of reactive oxygen species, sensing in the cell, a third redox stress pathway, now partnering up with the hypoxic stress response discovered at Dana Farber and the oxidative stress response. What is striking is that these look so similar. They all are centered around the cullen ligase. They are all regulated by oxidation of either substrate or E3, and they all produce a really intriguing output that is linked in some way to the particular stress condition that they are sensing. So I think this shows that this approach here, this design of the pathway, has really co-evolved multiple times to deal with very specific aspects of reactive oxygen species management. There's a lot of work that comes out of that. We're very, very excited about this. There's regulation. Are there other aspects of metabolism that fuel into this reductive stress response? We certainly believe that is true. We have now generated knockout mice in the system so that we can ask, how is this mitochondrial tuning or this rheostat used to control development? Can we tune it to our advantage in disease? And most importantly, this is, I think, going into the direction of inhibiting it to treat lung cancer that is caused by a KEAP1 mutation. That brings us to the last part, something that we're very excited about. Can we translate these into new approaches for drug discovery, both in terms of building small molecule degraders, but actually also just right away inhibit it or activate it. And so with this, I would want to close. I close by, again, thanking really the two true superstars of this. They had shown the courage to address difficult questions, the ingenuity to come up with their own model systems to build it, and then the persistence to go through all the hard years that you have, the difficulties to come up with beautiful models at the end. And I'm very, very grateful for both of them. Andrew Manford really is the leading person behind the reductive stress response, with lots of help from Fernando. And Elijah Mina discovered, and at the end solved the crystal structures as well of the dimerization quality control pathway. We could not do this without many collaborators that teach us, that inspire us, that help us. In this case, particularly John Corey and Eva Nogales and Basil Gerber in Eva's lab for Elijah's help, as well as Richard Harlan. And then I left Dan out here, Dan Nomura and Dennis Tito, for recent help with Andrew Manford's project. And of course, the funders as well. And with that, I'll thank you again for the invitation, and I'd be happy to take any questions in case you have any. Thank you.
Thank you very much. This was an inspiring seminar. I think we have time for 2 or 3 questions, so I'm just going to read them off the Q&As. If you have posted them to the chat, please repost them to the Q&A. I'm not going to look at the chat. So the first question we have is, how is formation of homo and heterodimer of BTB regulated? What is subcellular localization of formation of homo and heterodimers?
Yeah, so this is beautiful work coming out of Bernd Buchhaus' lab. So normally the cell actually forms BTB homodimers co-translationally to prevent the stochastic formation of heterodimers. And so heterodimers must come about mostly probably as a mistake that's made even in this, either in this co-translation process, or that you have a low extent of complex dissociation. However, because you have so many, even if only very few actually dissociate, you will actually produce quite a few. Another way of very actively making heterodimers is by overexpressing BTBs, and actually a lot of BTBs are overexpressed in cancer, suggesting that this is a pathological way of dealing with that as well.
Thank you. The next question we have is, would it make sense to ask whether the cell-autonomous mutations that cause defects in myogenesis can cause similar defects in a paracrine manner? For example, in co-culture experiments as a model for, say, cancer cachexia.
Yes, absolutely. I mean, absolutely. I mean, we know that especially with respect to, we know that myogenesis in the system is extremely sensitive to changes in mitochondrial output. That's what allowed us to discover this system in the first place. And we know that, mitochondrial activity is regulated by metabolism, and in many ways by the niche that is built out in this. So either co-culture, or more excitingly for us right now, organoid experiments, I think, would be a very, very intriguing way to study that pathway. And I believe that, you could build very innovative cool screening platforms to find new biology as well. That's a very good point, yes.
Maybe one more, maybe 2 more. Great work. Most of effects you have shown by FNIP1 are also regulated by AMPK. Have you checked if AMPK activation in cells mutant for FNIP1 can rescue the defect?
It cannot, yes. I mean, this caused, initially we went after both the AMPK link and the mTORC link because they had been published before, and we could not find anything that was genetically linked to this pathway that had anything to do with AMPK or mTOR1. That being said, we believe that there is crosstalk, of course, between those systems. It's not in the way that AMPK controls FNIP1. It might more be in that the output, if you degrade FNIP1, you produce ATP. That's what this pathway is doing, and that is regulating AMPK. So there's definitely crosstalk. We don't fully understand it, but it's not in any way linked to the core mechanistic things that I showed today.
I do have final questions. Great talk as usual. How conserved do you think these types of rheostats are for other types of non-redox based stress? For example, the titration of heat shock factors by chaperone complexes.
Whenever you have a system where making too much of one thing causes problems on another side, you will have such a rheostat. That would be my prediction. So I think they are actually going to be quite prevalent. This is the first time that we've discovered it, and we could build on an insane amount of work on the oxidative stress response to do this. So I assume that we can discover them, but I cannot predict yet how we discover them. The heat shock response is one of our most exciting examples where we try to find those things. Yes. So whoever you are, thank you for your comment and for your question.
Thank you very much, Michael. This was great. We're running out of time here, so just thank you again.